Georgie is proving the mainstream cancer model wrong. That no, estrogen is hurting, contributing to cancer. And by doing the exact opposite of what mainstream is doing for treating prostate cancer, he's seeing tumor reversal with the compounds that mainstream is saying is actually causing it. So they actually have it completely opposite.
But this is not news. Basically, Ray wrote 30 years ago, there's an article on estrogen and prostate cancer, and in that article, he has a sentence explaining how estrogen is really driving the cancer metabolism and how testosterone opposes estrogen in most of its effects, right? Hey, how are you?
You got a fresh shave today.
Yeah. I had to do some pictures for the school for the kids.
Oh, that's great. We all got the color code today. Shades of
Oh,
green.
yeah. The green. Yep. 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 I like green.
Georgie, how is everything going?
It's about the same. I don't think much has changed since the last time we spoke. I guess one thing is the DC budget is getting cut.
Yeah.
Or at least the new one is not getting approved. So of course they're not going to cut anything except police services. I mean, and then of course crime is ticking up, and people here are afraid it's going to be a repeat of 2020, like 2021, when we had the riots and everything. But we'll see. It's not there yet, but that's the worry for this summer. And there's also the housing market crash locally, at least, because of all the people that are being laid off from the federal government.
Yeah.
They're selling their houses and moving out, which is, I think, great news for the younger generation because they can finally afford a house. Not yet, but it's getting there.
Yeah. Yeah, because you're still in the DC area, right?
I live actually in DC proper.
Okay. Wow.
Yeah.
Wow. So you're starting to see the changes that are happening from the federal government in your immediate area.
Yeah, everything around here revolves around the federal government.
Yeah.
Any change there, especially financial, immediately reflects everywhere. So, there's of course also because of the layoffs and the downsizing, some federal agencies are getting closed completely. So this means more commercial real estate is empty now, and the DC government depends almost entirely on commercial real estate property taxes. So that's why the budget is getting lower, and they decided that they're not going to cut anything except the police. I don't know how this is going to work out, but we'll see. It'll be an interesting summer.
So I know that that's impacting, like the federal budget cuts is impacting research studies throughout the country. But your research studies are self-funded, which is
Yeah.
still wild to me. And I think that's an important thing for people to realize is like, Georgie is doing these cancer studies out of the kindness of your heart and interest in science. So
And also out of fear of the federal funding process, which I spent 15 years as a government contractor in writing proposals.
yeah,
And I don't want anything to do with the whole proposal process because that's how it works. You write a proposal and you get money from the government. There's always strings attached.
oh yeah,
And in general, it's a crappy environment. You don't have much latitude to do work on the ideas that you want to work on. So
no, in
for as
grad
long as you
school,
can maintain independence, there's something about the old American saying about truth that those who are willing to give up a little bit of freedom to give up freedom for a little bit of security that deserve neither, right? So at least as far as you're either doing your own work, your research work goes, that's definitely true. Like, don't give up freedom because very quickly you'll find yourself in a situation where you will not be working on your ideas, but somebody else's ideas, and you will also be told what to do.
in grad school, it was very like I didn't know this before going to grad school. And then in grad school, helping my advisors write research grants, I was like, oh, wouldn't it be so cool to study this? And they're like, that won't get funded. We can't do that.
Exactly.
You have to
Yep.
talk
Yep.
about this because this
Mhm.
we will get funded here. This is
Mhm.
what the government is interested in. And that's important because advisors require funding from federal agencies. Many of them do. And if they don't get that funding, then they don't have job security. They don't get
Mhm.
tenure.
Mhm.
They can't be a professor. And so you have to write specific grants tailored to what these organizations kind of want you to do. And so that's another important thing in the context of cancer managing cancer and managing diseases is a lot more profitable than preventing or curing them.
Yep.
And I
Yep.
think that that's one of the reasons why I'm so excited about Georgie's studies on cancer, because you're not reliant on these external funding sources so you can study what you want, and you're actually pursuing what is actually causing cancer? What can we do to actually reverse it?
Mhm.
So briefly, let me just try to summarize our last few podcasts where we've talked about your cancer studies, and you can come in and correct me at any point. But essentially, it kind of goes back to this bioenergetic view of cancer, which is when energy metabolism has failed, we cells then rely more on, glycolysis, cancer metabolism, the Warburg effect. And so in our last few podcasts, we talked about how the body needs energy to survive. And so we don't just have one pathway for energy production. We talked about in the last podcast, we have like a 6-lane highway where the first lane is the most ideal energy pathway. If that gets shut down due to a crash or an obstacle, then we can go to the next one, then the next one, then the next one. Well, cancer metabolism, hey, we are only on the last straw, and the bioenergetic view is, coming off of the work of Gilbert Ling and Warburg and Dr. Ray Peat, discussing that, hey, this is maybe failed energy production. And so if we can
Mhm.
instead turn back on proper oxidative phosphorylation, reverse states that are relying on glycolysis, maybe we can actually cure cancer. Is that
Yeah.
like a rough summary of kind of what we've talked about in past podcasts?
Yes, and also to point out that the glycolysis it's very important for things like wound healing. And basically, we whenever we cut ourselves, we have some kind of an injury, the cells in the area immediately kind of like temporarily shut off their oxidative phosphorylation, go back to glycolysis because when they in glycolysis, they can divide and grow very quickly. So, in other words, to fill up a gash wound or try to create new tissue, and that's an indispensable part of being alive. The problem is that whenever you're filling up wound with specific tissue, it's just cells without much differentiation in them. So once the initial process of healing the trauma is complete, you need to turn off the process of glycolysis because all we can do is divide and grow. In fact, the cancer metabolism, right? So this new tissue needs to be differentiated into the local tissue, whatever that is, muscle, tendon, bone, whatever. And in order for this differentiation process to occur, then oxidative phosphorylation needs to be turned back on. And there, as a result of the oxidative phosphorylation, you're producing differentiating hormones such as pregnenolone, progesterone, DHA, testosterone, etc. And those take care of kind of like calming down the newly created tissue, which is very much cancerous metabolically, and converting it to a normal tissue. Now, using the analogy of the wound, if you have an area of your body where you basically it's under constant assault somehow, whether it's radiation or polyunsaturated fats, whatever it is, over time, basically every time there is an assault on the local healthy tissue, basically it turns off its oxidative phosphorylation metabolism a little bit. But if this continues unopposed over time, the cell basically decides, literally says, well, it's too expensive for me to maintain the mitochondria. I need to focus my resources on surviving. So basically, it will dismantle its oxidative phosphorylation apparatus. It can completely destroy almost all of the electron transport chain complexes inside of its mitochondria. So basically, when this assault continues for a while, eventually the cancer cell may get to a point where all it can do is glycolysis. Now, there's a debate, and I recently was on another podcast with Dr. Seifried, who is the one who demonstrated that it's not the mutations that are causing the cancer, it's the dysfunctional mitochondria. And his research as well as mine shows that you can reverse a cancer cell and make it normal again, but only in the circumstances when the mitochondria is not damaged completely. If it's severely damaged, then the cell can and should be forced to commit apoptosis, which is the controlled self-destruction. And it looks like aspirin, which we discussed last time, or at least mentioned it, is very good at doing that. There's an old study that I shouldn't say old; it's about 80 years old, that I posted originally on the Repeat forum, showing that just a single tablet of aspirin daily, 325 milligrams, when it was given to animals that had cancer, basically went, I mean, circulated around the body when the aspirin was ingested, And then in the cancer tissue had the effect of switching off the cancer metabolism for the cells that were still relatively normal, and made them switch back to oxidative phosphorylation. And for the cells that were too damaged to actually do that switch, it somehow commanded them to commit apoptosis. That's really, that's really what we're looking for in terms of treating cancer. There may be a situation where the damage is too big and there needs to be I don't want to. I don't want to call it kill the cancer because that's the official medical dogma. The cell can actually recognize when it's too damaged to continue contributing to the organism when it's becoming a drain, and will commit apoptosis. But there are certain factors inside the cell after it's become cancerous that are preventing it from doing that. And one of the biggest factors is the pH factor. So all cancer cells are highly alkaline, and in order for them to commit apoptosis, they need to become acidic. Well, guess what? Aspirin is an acid. So when it gets inside of the cell, it just may succeed in dropping the pH to the point where the cancer cell can decide, okay, am I too damaged? Do I need to commit suicide, which is the apoptosis, or should I switch over to the oxidative phosphorylation metabolism and become a normal cell again?
Before we move on, I think it's important to clarify one thing. So, glycolysis, that's the backup pathway for energy production. It's needed in some parts of the body. We're all performing it right now to some degree, but in cancer, that gets massively upregulated. And it's important for people to understand that glycolysis occurs outside the mitochondria. So inside your cell, you've got a ton of different mitochondria. Glycolysis occurs outside of the mitochondria. It produces less ATP and it produces harmful byproducts such as lactic acid and things like that. So glycolysis is outside the mitochondria. Oxidative phosphorylation, which is the ideal energy production pathway, is when the energy substrates enter the mitochondria and go through the electron transport chain. What Georgie was referring to earlier is that some cancer cells, that pathway, the electron transport chain, is so damaged that the cell can't do oxidative phosphorylation anymore. For those cells, it's ideal that they commit apoptosis and die because they can't rebuild the machinery required for oxidative phosphorylation. And you're saying that aspirin has been shown to help with that process of helping the cell determine, hey, it's time for suicide, like cell death. And they do that by changing the pH. Aspirin
Hmm.
does.
Hmm.
Cool.
Yeah,
Okay. So
aspirin
that's
drops the
like well
pH.
established.
Yeah. So if a cell is too damaged, it will actually very quickly commit apoptosis, but it requires an acidic pH. And you can do this through several ways. Aspirin is one way. Dr. Pete's suggestion to breathing carbon dioxide is another way. Carbon dioxide is a so-called Lewis acid. Other compounds that are much more acidic than aspirin can also help, which is the 2,6-dihydroxybenzoic acid that I'm using for the cancer trials. That's actually one of the main reasons I started using it, because I was talking to Dr. Pete after one of the Danny Roddy shows, and I was asking him, okay, I know your position on aspirin. I know you said 6 grams daily for people with severe cancer. Um, is there anything that can be done to aspirin? Because I at that time I was already working with other chemists. Is there anything that can be done to the aspirin molecule to make it more effective? He said, "No, I want to touch the aspirin molecule, but if there was a molecule that was just like aspirin, but much more acidic and much more lipophilic, which will help the uptake into the cell, then I would go. I think that would be a good either adjunct or even a better version. Better option than using aspirin." So I did some searching and found out that 2,6-dihydroxybenzoic acid, which is a metabolite of aspirin, by the way, has these properties. So it may very well turn out that the benefits of aspirin are not from the aspirin molecule itself, but
The
through
metabolite.
its metabolism into these further more active metabolites down the line. And I want to go back a little bit to the topic of glycolysis. So there was this cancer researcher, very famous, I think, from Anderson Cancer Center down in University of Texas, and he gave a speech about 10 years ago, and he said we shouldn't think of cancer necessarily as a disease. We should think of it as going back to a more primitive version of life. So if you look at the cell basically has the cytoplasm, right, and you have the mitochondria inside. Current evolutionary theory says that this was actually a symbiotic relationship, that mitochondria are originally a different organism, possibly bacterial, that merged with the primitive human cell, which was only capable of doing glycolysis. And then basically this symbiosis allowed the cell the life to become more complex, because the energy produced by the mitochondria, which was originally a foreign organism, allowed life to basically acquire much more complex differentiating shapes. If you remove that symbiotic relationship, if you remove the mitochondria, then you go back to the primitive original cell that all it can do is glycolysis, right? And the energy produced from this primitive energetic pathway is only sufficient for division and growth. It's like a colony of bacteria or amoeba or fungi. All they can do is divide and grow and consume everything around them, including themselves. So really, that's what cancer is. If something is interfering with the symbiotic relationship of mitochondria with its host, and this could be many things. Any assault anything that injures the cells directly, physically, but also chemical assaults like the polyunsaturated fatty acids, estrogenic endocrine disruptors that are all around us, ionizing and non-ionizing radiation. I emphasize also non-ionizing because recent studies have shown that it's actually also pretty dangerous, especially when we're bathing in non-ionizing radiation 24/7 because of the phones in our pockets. Blue light. Whenever you live in a modern dwelling, you're probably under fluorescent lights. I am right now because those are the only ones available in the office. They don't give you the option of anything else. I mean, in the winter, I have a red light here, but it's something that I brought. The only option if you go to a commercial real estate building is you're gonna be basking under fluorescent lights. Hospitals, right? Childcare entities. Wherever you go, these lights are everywhere. And the reason they're ubiquitous is that they're cheap and they last long, right? But also at the expense of damaging our mitochondria. Our mitochondria does not like blue light because blue light is toxic to bacteria, viruses, fungi, all these primitive microorganisms, which is probably another corroboration of the theory that mitochondria is originally such an organism that fused with our primitive human cells, because blue light is toxic to mitochondria as well. You can actually get skin cancer just by doing the UV light, which is very heavy on it's a bit beyond the blue spectrum. But even blue light itself, if you sit under it for hours a day, at the very least, you start getting things like headaches, cognitive disturbances, potentially mental health disorders, and ultimately, of course, cancer. Because like I said before, if you interfere for sufficiently long periods of time with the symbiotic relationship of the mitochondria with its host cell, eventually the whole cell says, I don't want to maintain these entities here that cost energy to maintain and energy to continuously renovate, right? Which is the mitochondria. They're not functioning properly. They're a burden. So I'm going to start to dismantle them. And that's what happens over time whenever we're exposed to something that interferes with the oxidative phosphorylation process, which is, of course, entirely done in the mitochondria.
So, okay, glycolysis outside the mitochondria and then oxidative phosphorylation inside mitochondria through the electron transport chain. And it's important for people to understand that there's a lot of processes and complexes and steps along this chain. And there are various cofactors such as the vitamins and the minerals that we consume in our food, right? So the whole goal of metabolism is to take the energy that we consume in our food and then convert it into cellular energy. So it's a energy conversion process. So we want to convert high energy electrons into ATP for our cells to use, and we need various cofactors and vitamins along the way to do that. So Georgie's previous studies have shown that, having aspirin and then the metabolite of aspirin can help restore that electron flow, but also various B vitamins such as vitamin B1 and vitamin B3 can help restore energy production because maybe those were missing cofactors. Maybe those cancer cells didn't have enough of those very important vitamins, which are critical cofactors to this oxidative phosphorylation process. So you have recent updates on your studies, your cancer studies. What have you recently been investigating and what have you found for improvement of restoring oxidative phosphorylation?
So the screenshots that I sent you, I guess last week, they were on the breast cancer study and on a prostate cancer study. These two combined are probably responsible for the majority of cancers in both men and women in the United States, actually most of the developed world. And specifically, the breast cancer study is on the estrogen receptor positive cell line known as MCF-7. It's one of the most studied cell lines out there. More than 80% of the new cancer diagnoses are estrogen receptor positive. And I just wanted to do a study with it because it's such a relevant disease for all women around the world. And so far, it looks like basically there were several groups in the study. One with the combination of B vitamins plus aspirin, and then there's one with just with the metabolite of aspirin, 2,6-DHPA, and then there's one with pregnenolone. And there were because of previous studies with the B vitamins and aspirin and 2,6-DHPA, that's why I had these two groups. Pregnenolone comes from I haven't mentioned this anywhere, but about 4 or 5 years ago, this lab that I'm currently doing the studies with, we did an in vitro study with them with the MCF7 line and pregnenolone. And pregnenolone did the best compared to all of the other standard treatments, such as tamoxifen, clomiphene, aromatase inhibitors that were basically currently used at the time. So not only it was more effective at lower concentrations, but unlike all the other chemicals that were used, pregnenolone didn't cause any cell toxicity. There was no cell rupture. There was no leakage of cellular internals, including the mitochondria, into the environment, into the buffer. The lab said we think that pregnenolone, based on the results, should be able to treat breast cancer, the MCF7 line. We used the same cell line back then. So I kind of forgot about that result because I was dealing with the other in vivo studies with with the leukemia. I'm sorry, the mantle cell lymphoma and the prostate cancer, because the ones that I've been starting to do recently. But then I remember about this, and I decided to do a study with the MCF7 line again, but this time in vivo, and that's a human tumor just transplanted on an immunocompromised host, in this case, mouse. And then the three groups are aspirin plus the B vitamins, the 2,6-DHPA, and the pregnenolone. And so far, the tumor growth has stopped in all of the animals. And over the last 3 days, we started seeing regression of tumor growth. So the tumors is starting to disappear, and no sign of toxicity in either of the three groups. So we'll see what happens. I mean, based on the previous studies that I did with the mantle cell lymphoma, we're hoping for a complete regression of the tumor, which will be, I guess, according to medical standards, will be considered full remission. But then you have to stop treatment and see if the tumor recurs. And we did that already with a mantle cell lymphoma, which is why I call that one a complete cure, because after the tumor completely disappeared, we stopped the treatment, kept the mites alive for about a month, which is probably the equivalent of several years in a human, and the tumor did not recur. So the lab said, study's complete. We don't want to continue feeding these mice and doing nothing with them. So they moved them to another study. They didn't kill them. And then we're hoping for the same thing to happen with the breast cancer study this time. Same process. If the tumor completely disappears, we'll discontinue for 30 days. And if everything's fine, we'll call it a day and potentially publish, because we have an in vitro and an in vivo study with the same cell line. And the in vitro study compared one of the treatment groups in the in vivo study, which is the pregnenolone treatment, to all the other standards of care treatments that are currently used for this breast cancer. So it holds more weight, because all the other studies that I've been doing, is there's usually no. Actually, I do a couple of studies with control group just to see how quickly the animals die without treatment. And then after that, I only have active groups, in other words, the treatments that I want to, because there's no point paying for self-funded studies. I don't want to pay for a control group every single time, considering that it's pointless. They all die without treatment. I'm working with some of the most lethal tumors known to medicine. Aside from the breast cancer one, which is already pretty lethal if not treated, I'm also working with a pancreatic cancer cell line and a neuroglioblastoma, which is a brain tumor. And all of these are invariably lethal, and they kill the host within a couple of weeks without treatment.
So I think what you just said is incredible. The fact that all the groups performed the same, if not better, than standard care of treatment for cancer on
Better.
the
No, no,
better.
definitely better. So in other words, pregnenolone was effective in the in vitro study at lower concentrations compared to the established treatments. And unlike the established treatments, which had some toxicity, and that's known, nobody's going to deny that in medicine, that these molecules, clomiphene, tamoxifen, anastrozole, letrozole, they all have some toxicity. Pregnenolone did not have any. We did several, I think 2 or 3 different cell toxicity tests, and there was no leakage. There was no lactate dehydrogenase increase. There was no mitochondrial debris into the buffer. So basically, the cells looked normal. I mean, other than being cancerous, the cells looked normal. It eventually reverted to a normal metabolism. That's the other key thing. They didn't die, Pregnenolone was capable of converting their metabolism back to normal, which is the non-cancer metabolism.
Okay, let's dive into this a little bit. So, I think that it's well known for many people, especially in the bioenergetic peat space, that estrogen dominance is a huge major crisis that we're
Hmm.
all facing due to pesticides. Pesticides are known endocrine disruptors.
Hmm.
They lead to estrogen dominance. Phytoestrogens, those are plant compounds in like soy and flax. Those are ever increasing in our food supply. Various plastics that we're consuming. So we're being bombarded with these endocrine disruptors that are
Hmm.
leading to this, wide scale estrogen dominant state. So, estrogen dominance, can you speak on briefly how is that interfering with oxidative phosphorylation, so proper energy production? And then why do you think pregnenolone specifically is combating that?
So estrogen is a known reductant. And speaking of again of mitochondrial metabolism, you want a shift towards oxidative state. Whenever you have a reductive state, which means an overbuildup of electrons, you start getting things like reactive oxygen species and even reverse electron flow. And when I mentioned earlier that the cell starts to dismantle its mitochondrial apparatus, one of the first signals for the cell to do that is a shift in the redox state towards reduction. So estrogen, it's having two hydroxyl groups and three double bonds is basically a reductant from the point of view of oxidation and reduction. So its presence inside of the cell has about the exact opposite effects of what carbon dioxide does, which has an oxidative effect. Even though there's no direct interaction of the carbon dioxide molecule with reducing agents, so there's no direct oxidation reduction reaction going on because the acidic nature of the carbon dioxide molecule, it draws electrons across the cell as it moves, as it diffuses out of the cell, and that helps speed up the oxidative phosphorylation reaction. Also, carbon dioxide and lactate are inversely correlated in the body, and the more carbon dioxide you produce, the less lactate there is. Lactate is a reductant. So estrogen has this effect of blocking directly the effects of the crucial glucose oxidizing enzyme called pyruvate dehydrogenase. So whenever that one is blocked, and it's blocked in almost all cancers that we know of, actually all cancers that we know of, then you start overbuilding up lactic acid, or actually lactate. And lactate is a reductant. So again, signal not only is lactate itself directly procarcinogenic because it helps build new blood vessels, helps the cell to grow and divide, right? But also it kind of poisons the mitochondrial electron transport chain complex. And the cell basically takes the sign of increased reduction as a signal that it needs to start dismantling its mitochondria. So in other words, if you are in a state of chronically overproducing lactate, you're going to end up with severe mitochondrial dysfunction. And it is seen in conditions precursors to cancer, such as, let's say, type 2 diabetes, or other chronic conditions that are characterized by high levels of lactate. These people are always fatigued, their muscles are weak, they recover poorly from wounds or from exertion. They can't really do much exercise because they're not producing much energy, and that's because of the lactate. Lactate is a metabolic poison, so to speak, if it builds up, up to a certain level. And estrogen mimics to a large degree the effects of lactate while also stimulating its production. Also, estrogen stimulates lipolysis, which is the supply of fat to the cells to use as fuel, but that supply is coming from your internal fatty tissues that we all carry. The problem with supplying too much fat is that, as we've discussed before, there's a competition between fat and glucose for oxidation. And whenever you oversupply fat, you turn off the ability of the cell to oxidize glucose properly. All cancer cells that have been looked at so far over-oxidized fatty acids, and they do uptake a lot of glucose from the bloodstream, or in other words, from the food, but they don't properly oxidize it because they're being prevented from properly oxidizing it by the excess of fat. And what happens is that when you have an oversupply of glucose that cannot be properly utilized, you'll end up with a lot of lactate, because lactate is the emergency byproduct of pyruvate, which is the result of glucose entering the glycolysis cycle. In other words, we always have these two nutrients, the macronutrients, that they're the only two things that we can turn into energy. They're
Well,
always supplied to our cells.
that's why people say cancer feeds off of sugar. And
It
it's
doesn't feed off of sugar
and
because it's
it's
not using it properly. It's
they
wasting it.
they misinterpret that
Yeah,
the carbohydrates are being funneled through the glycolysis pathway,
exactly.
upregulating lactic acid, making things
Yes.
worse. The carbs aren't being used properly, they're not going into the mitochondria.
Yes, so the carbs will always first start going through the glycolysis, but in a normal healthy cell, once they go through glycolysis, the end product is pyruvate. And pyruvate will then enter the oxidative phosphorylation apparatus, which is inside the mitochondria, and then will generate carbon dioxide, water, and ATP. But if there's something blocking the entry of pyruvate, which is again a product of glucose, into the mitochondria, then this pyruvate starts to accumulate. And in order for the cell to continue living, at the absolute basic, it needs its glycolysis to continue to be functional. There is a possibility of even glycolysis shutting down, and that's what happen. It will happen if there's a huge shift of the cell towards reduction, similar to the cancer cell, but even more extreme. So even cancer cells can die if you make them to reduce. So in order to prevent this from happening, because the cell wants to stay alive, it needs to shift the balance back into towards oxidation. And what that means is that in a state of extreme reduction, when it's overproduction of pyruvate and it cannot go into the mitochondria, you build up pyruvate, but also there's a buildup of something called NADH, which is the reduced version of the crucial cofactor NAD plus, which is used in the mitochondria. So when there's a huge buildup of NADH, the cell says, because without NAD plus, even glycolysis cannot work. So the cell says, okay, my mitochondria is blocked. I have a huge buildup of a byproduct here from glycolysis, which is called pyruvate, and I have a huge buildup of NADH, and I do need NAD plus. I need the oxidized version of NADH. What can I do to oxidize NADH back into NAD? Well, normally that process happens in the mitochondria, but my mitochondria is blocked. What do? Well, the only thing I can do is use pyruvate instead of oxygen and oxidize NADH back into NAD plus, but the output of that process is lactate. And the only way this you get out of this vicious cycle is if the mitochondria starts to work again, number one, or if the cell decides that it's too damaged and basically commits apoptosis. And that's really the only two things that we can that can be approached therapeutically in cancer. This whole idea of killing the cancer cell while that cell is still very much active and viable, I think it backfires terribly, and we see that because once you start killing cells that are relatively healthy, and they basically they spill their entrails, so to speak, into the bloodstream. Multiple studies have demonstrated that this is a signal that becomes systemic, and the entire body decides that it's under attack. A lot of the mitochondrial debris and the debris from the nucleus trigger the exact same pathways and receptors that bacterial or viral infections trigger. So of course, when that happens, when you have an infection, you're under chronic inflammation because that's one way the foreign pathogen is being killed. But the problem with inflammation is that it also turns off systemically the oxidative phosphorylation metabolism because the body needs to focus resources on fighting pathogens. So when you kill the cancer cells that are not yet damaged enough that they really deserve to die, when you kill all these healthy cancer cells and they spill the entrails into the bloodstream, your entire body becomes inflamed, so it becomes a systemic issue, and the entire body says, I'm under attack, so I'm going to start turning off the oxidative phosphorylation everywhere, not just at the tumor site. So now you have basically your entire body capable of supporting new cancer cells. And I think that's really what explains the metastatic process. Once they cut off the tumor and blast that area with radiation and chemotherapy, and they spill all of these dead cells into the bloodstream, the entire body becomes basically shifting towards the cancer metabolism. And within a few months, you will see metastatic nodules prop up everywhere, disseminated. And that's the fear of every oncologist is that, yeah, we know very well how to kill almost every single primary tumor. But it's not a problem killing it. It's the problem that it comes back with a vengeance. And that it's no longer localized. It's no longer the spot where we initially killed it. Now it's like an army of a million little cancers and it's all over the body. And the bioenergetic theory says, well, of course, by killing this tumor, which should not have been killed, you should have tried to first coax it back into, acting normally because it is, at its base, most cancer cells are still structurally normal. They're metabolically deranged, but they're still normal. Some of them are too damaged, but it looks like it's a small percentage. So really the goal should be don't immediately go on an all-out assault, right? Which it turns out to be self-destructive. Try to coax the cancer cell to act normal again, and that can be done metabolically with various approaches. Because when you attack it, basically you tell the body something is trying to kill you. It really is. I mean, no doctor will deny the fact that ionizing radiation can kill you if it's in a too high of a dose, right? Chemotherapy, especially the cytotoxic kind, almost all of them are. It's also basically they're trying to kill the tumor while trying to limit the damage for the rest of the cells. But guess what? You cannot limit the damage for the rest of the cells. Why? Because there's very little difference between a normal cell and a cancer cell unless it's too damaged. It's just their metabolism is a little bit different. But if you assault truly the cancer cell and try to kill it, you're telling all the other cells around it, because they constantly communicate. You say something is trying to destroy, literally destroy part of the body here. And the body says, okay, then I'm going to mobilize my entire resources and shift them towards the cancer metabolism, because that's the metabolism for recovery, for building up new tissue. And that's why you get, metastatic nodules everywhere if you go on an all-out assault against the tumor in any specific organ.
I think something that you said that was funny to me is you said the words healthy cancer cell. And so that's just like so opposite of what mainstream thinks, but that is such a hopeful view that, hey, that cell has cancer metabolism, but it's still has the
Structurally healthy.
machinery,
Yes.
it can
Mhm.
get turned back on, energy production can get turned back on, and it can exit cancer metabolism, and that pyruvate, which is a product of glycolysis, can re-enter the mitochondria and go through the electron transport chain. So what about so estrogen dominance interferes with pyruvate entering the electron transport chain through PDH.
And
So
also, it's a reductant itself, so it basically tells the cell that, oh, I need to start dismantling my mitochondrial apparatus, because in a state of reduction, the mitochondrial apparatus does not work very well. It does not reoxidize NADH back into NAD. You do not produce sufficient amounts of ATP without the oxidative apparatus. So all of this machinery in the cell requires ATP to maintain and to continuously rebuild. So if you're not producing sufficient amount of ATP, which you're not, if the mitochondria is not working, then the cell is saying that I don't need that apparatus, so I'm going to get rid of it.
so what about pregnenolone? So like, okay, can you explain pregnenolone is a downstream of progesterone
No,
or
of
no,
cholesterol.
upstream.
Yeah, upstream. Yeah,
So
it's the
what
mother, the
about
mother of all hormones. If we can use this, it's not the mother. They call it the grandfather.
the
Then the whole
grandfather? The
progesterone
grandfather.
is the mother.
So what about pregnenolone is improving the estrogen dominance
Mhm.
locally?
Yes. So first of all, pregnenolone, progesterone, to a smaller degree, DHEA, testosterone, dihydrotestosterone, they're all aromatase inhibitors. So they directly limit the synthesis of new estrogen, no matter where it is being synthesized. Every cell expresses the aromatase enzyme, which is the rate limiting step in synthesizing estrogen. So anything that inhibits aromatase, uh, basically will decrease the production of estrogen. Unsurprisingly, aromatase inhibitors are some of the standard of care drugs for treating the estrogen receptor positive breast cancer, which is why I mentioned that we compared it to pregnenolone in the MCF7 in vitro study. We compared it to letrozole and anastrozole. Both of them are classic, I think, second generation aromatase inhibitors, very potent, currently standard of care treatment for estrogen receptor positive breast cancer. So anything that acts as an aromatase inhibitor will help decrease the estrogenic burden inside of the cell. And also outside of the cell, because the cell gets estrogen through two methods. One, synthesize it locally inside, right? Two, get it from the bloodstream from other tissues that are synthesizing too much. And cancer cells do that, right? I guess a bit of a side note, that's a great bystander effect. Cancer cell overproduces estrogen, some of the estrogen seeps out, goes to other cells, right? Has the exact same pro-cancer effect, maybe not to that degree as on an already formed tumor, but it's definitely priming the other cells to be less oxidizing and more reducing. In other words, getting closer to the cancer metabolism. So you want ideally two things. Number one, limit the local synthesis of estrogen, which is what an aromatase inhibitor will do. And number two, limit its uptake externally from the bloodstream into the cell. Uh, Several studies demonstrated that pregnenolone, progesterone, and testosterone and dihydrotestosterone interfere with the uptake of external estrogen inside of the cell. So with sufficiently high doses of pregnenolone, you should be able to achieve a significant degree of both. In other words, limit the production of estrogen locally. Maybe I don't think it's going to you manage to completely abolish it, but limit it, because in a tumor cell, in a cancer cell, it's definitely upregulated. And also, if the cell is relatively healthy, but there's a tumor somewhere else producing estrogen and distributing it through the circulation, limit the uptake of that external estrogen into the cell. Pregnenolone does both. So does progesterone.
So far your studies have really shown B vitamins, pregnenolone, aspirin, and the byproduct of aspirin have really been helpful.
Yep.
Have you published anywhere what the equivalent dosages would be from your mouse
Yeah.
studies to humans?
So let's see. I think the current dose for the pregnenolone for the mouse study is 200 milligrams per kilogram daily. So that's, let's see, it's about 15 milligrams per kilogram for a human
50
pregnenolone
or
daily.
15?
15, 1 5.
50.
Yeah, 1 5.
Okay.
So that means probably over a gram for an adult. But again, no toxicity, right? Dr. Pete mentioned when he was very sick, I think back in the 60s or the 70s, he spent months in bed, and he recovered miraculously after something told him to try pregnenolone emulsified into vitamin E. And like he said, after that, he, for several years later, he was using several grams daily. So the unique thing about pregnenolone is that it seems to only work if there is an imbalance of a steroid further down from it, somewhere down the steroidal cascade. So if everything is fine, pregnenolone will be largely inactive. It will still help stabilize the cell, because all steroids are lipids, and specifically highly lipophilic ones like pregnenolone. They make the cell very impermeable, much more lipophilic than the blood, which is hydrophilic. And most of the assault that a cell gets exposed to is coming from the outside. In other words, specifically the food or the endocrine disruptors, in order to damage a cell or injure a cell, they have to go inside of you, and then they have to be distributed by the blood to the cells. But in order to injure the cell, they have to be able to get inside of the cell. And usually, the more lipophilic the cell is, the more impervious is to entry of these, agents of attack from the outside, from the bloodstream. So pregnenolone, being highly lipophilic, actually helps the cell become more impervious to such attacks. And unsurprisingly, saturated fats do the exact same thing. They make the cell more lipophilic. Polyunsaturated fats, because of the number of double bonds in them, are much more hydrophilic, even though they're still lipids, but they're much more soluble in water than saturated fats are, which are almost completely soluble in water. So when you ingested a lot of PUFA, you make it, of course, they go to the cells and, infuse with them, diffuse inside of them. So this makes, over time, as the PUFA concentration inside of cell increases, makes the cell much more able, or actually much more vulnerable to assault from whatever is coming through the bloodstream, because that molecule will have an easier time getting inside of the cell and doing whatever damage it's supposed to do. So yeah, pregnenolone is the structural cell stabilizer, and if there is an imbalance somewhere steroidally, pregnenolone will take care of it.
And PUFAs interfere with the detoxification of estrogen in your liver.
Absolutely.
So there's
Yeah.
another assault.
Yeah.
So we're not only being bombarded by endocrine disruptors in our environment, our high PUFA intakes are interfering with our body's natural process of detoxifying estrogen, just making the whole estrogen dominant state worse. So we're
Yes.
saying.
And also, Dr. Pete said years ago, but I also found the studies that he was referring to, that the detoxification of estrogen, which mostly happens in the liver, requires vitamins B1, B2, and B3. So I don't have B2 in my current studies, but B1 and B2 are definitely present. So a side effect so these vitamins are not just acting as cofactors, even though that's probably their main role. They're also helping the liver of the sick animal detox all of this estrogen, which is upregulated in every tumor tissue, because estrogen is a growth promoting hormone. The tumor produces it because the tumor wants to continue to divide and grow, because in the absence of mitochondrial metabolism, that's the only thing the tumor can do. And because it's a collection of cells that are all communicating with each other and they act as a group, the group is protecting itself. It wants to increase its numbers, and that's why it keeps producing estrogen and lactate, both of them reductants, helping it grow until it basically takes over the entire body. So the vitamins help the liver of the organism detox the extra estrogen that results in a tumor-bearing organism.
Me? Oh, okay. Yeah, I had a question about the pregnenolone. So, if somebody's aware they're estrogen dominant and maybe on the path to developing cancer, or any issue in the future, the general recommendation a lot of people give in this community is to take progesterone. And so I'm wondering what the advantages of using pregnenolone alongside the progesterone, or if that's overkill, or if you would actually recommend that somebody does take some sort of maintenance dose of pregnenolone.
So I did Pregnenolone because I spoke to Dr. P. He said it's unique because it's inactive unless there is an issue, right? Progesterone is not. It's already a preformed hormone. There are receptors for it in the cell. It activates the progesterone receptor, right? We all know that. But it also blocks the estrogen receptor. It activates the GABA receptors. It's also an aromatase inhibitor. So pregnenolone, the real direct effect that it has is that it's a mild aromatase inhibitor, but it's largely inert unless there's an issue in the body. And unlike progesterone, pregnenolone is not a controlled substance in most countries. Progesterone is. We here in America are lucky that progesterone is still not, the FDA has not clamped down yet. Maybe it will, hopefully it won't. When you go to any other country around the world, progesterone is usually a prescription drug, and in some countries it's regulated not very differently than anabolic androgenic steroids. So, Europe is one example. DHA and progesterone, no. These are hardcore drugs there. So I wanted to do something that's more widely accessible to people, and that's one of the reasons I also did the B vitamins plus aspirin, even though the indication that the metabolite of aspirin 2,6-DHPA works better. But it's such a unique chemical. Oh, I was going to say unique, but it's a much rarer chemical and much more difficult to find. And in fact, you probably cannot buy it unless you run a company like me, or a doctor or a chemist or whatever, because they don't sell to individuals. So I wanted to do something that's accessible to everybody, even though they may be a better option. So I think pregnenolone is widely accessible, but as far as cancer is concerned, I think progesterone will probably be a better choice because it has some additional effects. First of all, it's a stronger aromatase inhibitor. It has a direct blockade of the estrogenic receptor as well. So if estrogen is already there, progesterone will actually prevent it from exerting its effects. It also limits its new synthesis, and just like pregnenolone, it interferes with its uptake from the cell. So it has an additional effect on top of pregnenolone, because pregnenolone itself does not act on the estrogenic receptors, right? So progesterone really covers everything that estrogen does, and it has some additional things such as estrogen activates the cyclooxygenase and the lipoxygenase enzymes. In other words, increased inflammation. Progesterone blocks both of these, right? I think pregnenolone blocks only one. So I think for if the cancer is already established, and anything over stage one, I think I'll go directly with progesterone. Um, and aspirin. These are not mutually exclusive. If it's for general preventive purposes, I think a lower dose pregnenolone, about 30 milligrams daily, which Ray also said. I've personally experienced this to work best for me. Somebody else consulted with Ray. He said that the multi-gram doses that he did definitely help, but he thinks the majority of that is wasted because there's a limit on how much pregnenolone he can absorb. And when that person asked Ray, what do you think is the optimal dose? He said, I think 10 to 30 milligrams daily, especially if combined with aspirin. So I think that would be what I would do. I don't want to say recommendation. That's what I would do if I felt, that I'm shifting towards the cancer metabolism. Aspirin and pregnenolone daily. By the way, I already do that.
So, can you talk about how the pregnenolone performed for the prostate cancer study that you did?
Yes. So, actually, the prostate cancer study is done with progesterone, not pregnenolone.
Oh,
Yes, because I wanted to show the main goal. Actually, two main goals behind the prostate cancer study. Number one, to show that androgens are not only not the cause, but a therapeutic, right? And number two, that estrogen is definitely a cause there. Now, the way you do this, you basically, because estrogen and androgens are antagonistic to each other. So you develop several groups, and one of them, I think the one that I like the most is basically using dihydrotestosterone, which currently is being labeled as the cause of prostate cancer. Talk to any urologist, any urological oncologist, and they'll tell you, yes, prostate cancer, initially at least, in its androgen receptive form, is definitely caused by dihydrotestosterone. That's why we're giving all of these literally chemically castrating drugs. In some cases, they still physically castrate people. And as part of the chemical castration, they also administer estrogen, right? So I said, okay, let's see if we can flip this on its head. Let's see what happens if I
Wait, hold
create
on.
this
They
prostate
they
cancer tumor.
administer estrogen
Yes,
for
ethinyl
prostate cancer?
estradiol, the exact same pill that is still given to women as part of contraception treatment or menopausal treatment. There's something called ethinyl estradiol. It's the most widely used synthetic estrogen in medicine, and in the body quickly gets hydrolyzed to regular estradiol, which is the most potent endogenous estrogen that we produce.
It sounds like their understanding there is like similar understanding to what they say about PCOS.
Yes. They said estrogen is causing it, but androgen is
Yes.
causing it, right? But there are multiple studies showing that the way to induce PCOS in female animals, you just inject them with a sufficiently high dose of estrogen. It just doesn't. And there's another study, women with PCOS who were given dihydrotestosterone as treatment. By the way, medicine claims that PCOS is also caused by androgens. So not only didn't dihydrotestosterone, which is the most potent androgen endogenous that we produce, both men and women, not only didn't make their PCOS worse, it actually treated it. And they lost weight, mostly fat, they gained muscle, their libido improved, their mood improved. So it looks like, really the androgen is not male specific. These steroids have beneficial effects in both genders. I recently posted something on that, basically that without androgens, female organisms and male organisms are sterile. The experiment was done with chickens, which you should appreciate. They created these genetically mutated chickens that were missing the androgen receptor. And basically, these chickens did not ovulate, did not lay eggs, did not like the males. They fought them, and they fought between each other too, right? But basically, they also confirmed the same thing with regular female chickens. They depleted their androgens. The androgen receptor was still there, right? And the exact same thing happened. By the way, the exact same thing happened in males too. So they removed their androgens. Of course, no sperm produced, no viable sperm. There was no copulation. The males lost interest in the females, and vice versa. So really, we're looking at a steroid that is systemically important to both genders. Yes, it does have some sexually dimorphic effects, right? But it's only in the higher doses. In the lower doses, it's acting primarily as a metabolic stimulator and a health promoter.
I'm sorry, I interrupted you. So you were saying that you were going through the standard of care for prostate cancer, which
Right.
involved estrogen, which is just so funny to me because estrogen is contributing to prostate cancer. So they're potentially making it worse. And your study did the literal exact opposite with progesterone treatment. So I
Right.
cut you off.
So they don't tell you that estrogen causes the cancer. They're saying it's a treatment for it. It's androgens that are causing the cancer. So we're gonna remove all the androgens from you. But guess what? If you're willing to do it, we'll do the physical castration. If you're not, then we'll do the chemical castration. And that's really that is the standard of care therapy for all newly diagnosed prostate cancers. 99% of which are androgen receptor positive. In other words, they're receptive to androgens. And medicine is saying it's the androgen that's causing the prostate cancer to form and to grow. Specifically, the androgen dihydrotestosterone, because it is the most potent androgen there is in the body. So they give you ethinyl estradiol as one form of chemical castration, and they'll give you another one called leuprolide, which basically blocks the pituitary from producing the pituitary hormones LH and FSH, luteinizing hormone and follicular stimulating hormone, without which the gonads are not going to get the signal to produce androgens. So that's really so in other words, the chemical setting by attacking both your pituitary and your gonads, because ethinyl estradiol or any estrogen in general suppresses the synthesis of androgens in the gonads of males. It also suppresses the synthesis of progesterone in the ovaries of females. So high doses of estrogen can actually chemically castrate both men and women. And something really funny happens when you administer high dose of estrogen. You may have seen it in modern humans, especially the city-dwelling ones. So whenever estrogen elevates, males feminize, but females masculinize. In other words, the sexual dimorphism all but disappears. And you can see that in very old people, where the males and the females are almost indistinguishable from each other, right?
Unisex.
Say it again? Yeah, exactly, unisex. And the reason for that is estrogen. The only hormone that does not decline with age, actually, aside from cortisol, is estrogen. Everything else, all of the youth metabolic promoting hormones, such as pregnenolone, progesterone, DHEA, androgens like testosterone and dihydrotestosterone, all of them plummet in both males and females. The two things that remain are cortisol and estrogen. Cortisol can actually produce all of the symptoms of aging in a very short period of time if you inject high enough dose. And estrogen removes the sexual dimorphism and destroys the oxidative phosphorylation capacity. So you're really basically just an old clump of cancerous cells that is just waiting to either outbreak and kill the host, but you're definitely not going to be young again as once you get into the stage. And it's not a surprise. It's not surprising that up until a few years ago, the rates of cancer were highly correlated with aging. In other words, the older you get, the more likely you are to get cancer. This is now strikingly, and I would say catastrophically, reversed. Or at least in the younger portion of the spectrum, people under 30 years of age now are seeing skyrocketing rates of cancer, especially cancers that were thought to be, like they used to call them old man cancer, lung cancer, colon cancer, liver cancer, prostate cancer. These are now skyrocketing in the youngest cohorts. So we know that something's happening in our environment that's definitely not due to mutations, because they're not going to affect just one age group, right? That's promoting the cancer. So back to the prostate cancer thing, basically. So they capture you by giving you estrogen, and something that blocks your pituitary, right? So I said, okay, let's do the exact opposite. Let's give the cancer promoter dihydrotestosterone, right? And since you claim estrogen is beneficial, let's give something that limits the production of estrogen, which is an aromatase inhibitor. And let's see what happens when we give these two in combination. They should be, according to medicine, they should really decimate these animals, because you're suppressing the estrogen, you're giving them extra androgens. I mean, the tumor should really become bigger than the animal. That's not what happens. In fact, like the group that is getting a combination of dihydrotestosterone plus the steroidal aromatase inhibitor known as exemestane in doses that are human equivalent to what is being currently given to humans for both of these, they're both steroids. Basically, at this point, in the group that's getting dihydrotestosterone plus exemestane, the tumor size is 0. So it started flat, then it started growing, then it started going down, and now it's at 0. So we're waiting to see if the scar where initially the tumor was implanted, because there's always a little bit of scar tissue, if that will disappear. That's a sign that the tumor is completely gone, and that we can stop treatment and see what happens afterwards. But as of right now, there is a full remission, because medicine doesn't like to use the word cure, in the group that's getting the exact opposite of what medicine says should be receiving. And there's some other groups, because I said, okay, DHT is a controlled chemical. It's considered an androgenic anabolic steroid, which it is. Okay, so what can we do that's more over the counter? Well, let's have one group that does progesterone, which is also anti-estrogenic, right? And exemestane, which is also anti-estrogenic. That group has also currently a tumor size of 0. And then there's various other combinations, such as basically there's progesterone by itself at a much higher dose, which is equivalent to the one I mentioned for Pregnenolone for the breast cancer. It's about 15 milligrams per kilogram daily. So again, over a gram for a human. But other than making you sleepy, I don't think what is going to cause any other side effects. And then there is a group that is getting only estrogen in the prostate cancer group, and then the tumor is growing up. So once the study completes, either they will shoot me or I will manage to publish it. And then at that point, I think it will be very difficult for medicine to continue as is, at least as far as urology is concerned, because it's the primary cancer they're dealing with. Prostate cancer is the most common cancer in males over, I think, a certain age, over 50 or whatnot. And it looks like so far that they're doing the exact opposite of curing it. They're probably causing it. And by the way, it's already been acknowledged. So there's something that they call shock androgen therapy, and they're saying, okay, we know that when we castrate the male, the tumor initially shrinks but does not disappear. And it stays there for, let's say, 10 years. And eventually, invariably, comes back as the highly aggressive, almost universally lethal, funnily named castration-resistant prostate cancer. So first they castrate you, they think you respond well, you really don't. Your quality of life is destroyed, obviously, right? Um, and then after 10 years, you develop a cancer that they really cannot treat. And that's usually the one that kills you. The castration-resistant one, which is, they claim it's insensitive to androgens because the castration no longer works for it. Yet, recent studies demonstrated that in those specific types of cancer that it should not be responding to androgens, when they injected the prostates of males that were about to die from this highly aggressive prostate cancer, when they injected their prostates directly with testosterone, which it should be causing the cancer, should be making it growing, completely made it disappear. So the way they got out of this was like, well, we're calling this restorative or like a rejuvenating androgen shock therapy. Something about the androgen snaps the castration resistant cell back into castration sensitive state, and then it continued to castrate you, and then they say, oh, now it's working. No, it's the testosterone that's really doing the benefit. So there's already evidence that this hypothesis of androgen driven prostate cancer is completely, I don't want to call it fraudulent because I'll probably get sued or worse, but it's definitely wrong. We're seeing the exact opposite.
I don't think people realize how amazing these last couple minutes, like, Georgie is proving the mainstream cancer model wrong. That no, estrogen is hurting, contributing to cancer. And by doing the exact opposite of what mainstream is doing for treating prostate cancer, he's seeing tumor reversal with the compounds that mainstream is saying is actually causing it. So they actually have it completely opposite.
Yeah, that's why I'm very careful about saying this because, and I will try to publish it because, if people listen to this podcast and realize that their loved ones are being harmed or potentially killed by giving them these harmful treatments, there'll be class action lawsuits and potentially even congressional hearings. But this is not news. Basically, Ray wrote 30 years ago. There's an article on estrogen and prostate cancer, and in that article he has a sentence explaining how estrogen is really driving the cancer metabolism and how testosterone opposes estrogen in most of its effects, right? And he has a sentence that says, in light of these effects of testosterone, has anybody tried treating prostate cancer with testosterone? Well, 4 years later, they tried treating terminal people with prostate cancer with testosterone, and their tumors disappeared. But it didn't cause any kind of a backlash or any kind of a collapse of credibility. The doctor simply turned around and said, "Well, because testosterone is the precursor to estrogen, when we inject it in the prostate of these cancer cells stricken of these cancer patients, it's rapidly converting into estrogen, and that's how it's actually helping for the cancer." Of course, just before that, they were claiming that no matter how you put testosterone into the male body, it will definitely contribute to the growth of cancer. That's why we're going to do everything possible to castrate you so there's no estrogen in your body. But then when we inject testosterone in a specific manner, now we get to claim that it's converting to estrogen, and that's how it's helping. So which one is it? Either it's always dangerous, right? Or here's the other part. When you inject testosterone into the prostate, it does not have high expression of the enzyme aromatase. It's actually one of the lowest. Its cells have one of the lowest expression of the enzyme aromatase. So very little of testosterone will convert into estrogen when injected into the prostate. The majority of estrogen, of the circulating estrogen produced by tissues, is done in the fatty tissue. They have a very high expression of aromatase, but not in the prostate. The prostate does express another enzyme, and it's called 5-alpha reductase, which takes testosterone and produces dihydrotestosterone. So their experiment effectively confirmed, years before I did mine, that dihydrotestosterone, which is currently claimed to be the cause of prostate cancer, is actually therapeutic. By injecting testosterone into their prostates, they massively elevated the levels inside of the prostate of the androgen dihydrotestosterone. All I did was just administer dihydrotestosterone preformed instead of administering its precursor testosterone.
I guess I'm just confused, like,
How
even with
so
I mean, like, even with just a basic understanding of the human body in males versus female, it would just seem illogical that estrogen would be the solution for a male. So, like, there's obviously funding or some corruption going
estrogen
on.
is the whole estrogen therapy, estrogen compound, it's so profitable for these companies.
Of course, it's the main cash cow to this day. I mean, it's used in abortions. Sometimes they administer the abortion pill, mifepristone, but it's very often also administered together with ethinyl estradiol and a prostaglandin to stimulate contraction of the uterus. And if they don't have access to mifepristone estrogens have been known since time immemorial to be so-called abortive agents. So people who were in the livestock industry, especially the primitive societies, if they didn't want their flock to like give birth, because they were afraid that there's going to be deformities or whatnot, they will take them to a place where they can eat either clover, or what do you call it, or members of the legume family, beans, soybeans, whatever, because these are highly estrogenic. And if the animals eat a sufficient amount of phytoestrogens, they spontaneously abort. So really, estrogen is an abortive agent, and in high enough doses, it can kill you because it has such a poisoning effect on oxidative phosphorylation. And just that alone should be enough to know that you do not want to mess with estrogen. You do not want to administer it externally. You definitely produce more than you need internally. And the fact that, look, if androgens were the cause of prostate cancer, then we should have 20-year-old males basically coming down with prostate cancer left and right, because they have relatively low estrogen and relatively high androgens. And in fact, there is something called the androgens to the estrogens ratio. So you pull together all the androgens, right? You measure the concentration in the blood, and then you pull together all the estrogens. And if that ratio is not at least 50-1 in favor of the androgens, multiple studies have demonstrated that males come down with a variety of chronic conditions, specifically liver disease, lung disease, infertility, obesity. So the androgen to the estrogen ratio is crucial, and it needs to be in favor of the androgens in order for the male to be healthy throughout throughout his lifetime. Somehow, at some point, specifically for prostate cancer, they say, no, it's the exact opposite. The high androgen to estrogen ratio is terrible for the prostate cancer. It's causing it. We need to reverse that ratio by removing all of the androgens, by castrating you, and simultaneously pumping you full of additional estrogen by giving you ethinyl estradiol. And all there's been, if this therapy was working, we should be seeing a dramatic decline in the rates of prostate cancer, or at least high rates of cure. That's not what's happening. No. So once you get prostate cancer, I think now the recommendation for most men is, because medicine has realized that it's failed miserably in treating it, unless your initial diagnosis says that you have the high grade, like the so-called aggressive castration-resistant prostate cancer, because you can get that from the very beginning, but it's very small percentage, maybe about 5% of the people of the initial diagnosis. So 95% initially get the diagnosis of the androgen-sensitive regular prostate cancer that most men get. The advice of most urologists these days is, don't touch it. Leave it alone. We're going to do watchful waiting. I don't know what it means. Like, it's waiting to do something spectacular. So watchful waiting, and if it doesn't grow, it doesn't cause problems, you're more likely to die of old age or something else than of this cancer, right? So I think they're trying to shift away from the aggressive treatment of this initial mild or slowly developing form of cancer, because they realize that castrating the males and giving them additional estrogen, which is also pharmacastration additional, yes, it shrinks the tumor for about 5 to 10 years, but invariably at the end of that period, the men come down. Basically, the cancer comes back with a vengeance, and now it's the highly aggressive, treatment-resistant prostate cancer that medicine doesn't even know how to treat. So eventually, it kills the males, and it becomes metastatic too. The initial prostate cancer stays localized to the prostate. So for most men, they don't even know they have it. Ray mentioned that I forgot what was her name. The Queen of Austria, I think in the 1800s, mandated that everybody who dies in a hospital in Austria has to undergo an autopsy. And I think that's the case to this day in most countries. But anyway, she's the one who started it. And then doctors quickly realized that every person over the age, I think it was 40, had already at least one diagnosable localized cancer, which was kept under control. So it's there, it's not bothering anybody. And every single male already over 35 had a diagnosable tumor nodule in his prostate. So in other words, virtually all males over 35 are living full productive lives with already formed prostate cancers that, unless you mess with them, are probably not going to cause much trouble, and you're likely to die of other causes or of old age before this tumor starts causing trouble. Now, of course, if you take aspirin, progesterone, pregnenolone, etc., even that tumor may disappear. But the moral of the story is that do not try to attack the still healthy cancer cell, because it will come back with a vengeance. If you make the environment inhospitable for a cell that can still revert back to normal, it will turn into evil reincarnate and eventually kill the host.
Okay, so to summarize, Mainstream cancer treatment believes that androgens are the cause of cancer. So we should,
Specifically one, DHT, dihydrotestosterone, which is the most potent one there is.
and so let's remove exposure to that and instead increase exposure to estrogen. Well, Georgie's cancer studies, they're not done yet, but the preliminary results have shown that no, the exact opposite of that is true, that removing the estrogen source and instead uh, exposing the cells to androgens actually reverse the tumor. And if the mainstream cancer model that estrogen is helping and androgens are harming, if that was true, then why are we seeing cancer rates on the rise? It helps you can better understand why cancer is on the rise if you understand that estrogen is causing and contributing to it because it's interfering with energy production. And if we take a step back and reflect on what our environment and food system exposes us to, we have drastically increased our exposure to estrogenic like substances, endocrine disruptors such as pesticides, phytoestrogens like soy and flax, which are now being fed to animals as well, plastics, and many other endocrine disrupting compounds in just the environment that we live in. So why would exposing us to more help with that? But it is a lot more profitable to manage chronic diseases such as cancer than it is to cure them.
Yeah. Georgie, I
And I
was
also want to
laughing a
point
little bit
out, not many
earlier
people
because
know, but in
genuinely
2000,
concerned for
oh, go
your
ahead.
safety at a certain point.
No, this
Like you're
the thing is
exposing all
the
this.
light here is a motion sensor and it's behind one of the cabinets, which you can't see right now. And basically, unless I shake the desk or do something else, it periodically turns off the lights.
Sarah just said that she's concerned for your safety.
Well, it's just something I was thinking throughout this podcast because I feel like you're saying information that's so important and
Must protect Georgie.
it obviously is something that they're not trying to get out there.
Yes, but I think it's getting there. They're just going to try to present it as like a paradigm shift.
Like
Dr. Seifert is
serotonin
probably,
and SSRIs.
yeah, yeah. So RFK Jr. is already running with that information. So if I think he's a much higher value target than I am. One of the reasons why I haven't published these yet is actually one is like a bit of a concern because I talked to several professors here who work in biochemistry, and also the chemical group in Bulgaria. They're all established professors. So they approached some journals, and the journal said it's probably not a good idea to run with this officially right now. And I said, why? Well, because it's such a so you should be ready with lawyers because they're going to attack, right? Number one. Number two, most journals are not going to be willing to pick this up and run with it. They will want at the very least the confirmation in a human trial, right? And then it has to be done under somebody very knowledgeable and famous. When you do something like that, you're basically inviting because the cancer industry is huge and massive, very powerful. They will immediately challenge the study, right? And then they'll say they'll counter it by saying, "Well, we did the exact same study in our lab, and it's not showing these results, right?" So they'll have the study what is it called, retracted. And also, once you have a study retracted, it severely limits your ability to publish in the future because it's considered like you did a fraud or something, right? And then most of the people, the professors that I'm talking to, are not willing to risk their careers publishing something like this, considering it's going to get challenged. So the best thing I can do right now is simply put this information out there. I already have confirmation from individual doctors that are treating their patients this way, and it's becoming a grassroots movement. So if it works for sufficiently large number of people, then I don't need to publish. In fact, I don't care about publishing. Why do I need to give the enemies a sign that we're brewing a resistance here and the resistance is working? So really, I don't see a point in publishing right now, but already doctors have picked up this research and are starting to use it on the prostate cancer patients and breast cancer patients and patients with other tumors, especially the treatment with B vitamins and aspirin. I'll forward you an email from a oncologist who's in Ireland, and he already has, I think, 7 completely cured patients, at least claims that their tumors are completely gone away and their surgeries have been canceled because the primary tumor is gone. So that's what I'm looking at. That's what I'm looking. That's my goal, really, to give this to the masses. And once this becomes like a prairie fire, too big to stop, that it doesn't matter if they come after me or not.
Yeah.
Every time we do one of these podcasts, we get emails for people asking for physician recommendations that maybe you are aware of, that are aware of your studies. Is that something that you can provide us maybe
Yeah.
after with the list for
Yeah,
people?
yeah. So not all of them are physicians, but one of them is he has a PhD in biochemistry. The other one is I think, doctor of osteopathy. Another one is a licensed nurse care practitioner, I believe. And then 3 or 4 are nutritionists. So I have like a list of 7 or 8 of them that when people email me and say, "Hey, I want you to do consulting for me on these health topics," I usually say, "I'm sorry, can't do for legal reasons. But here's a list of people you can contact." So I can send
That'll
you
be
the
Put
same
great.
list.
that in the description. No, well, yeah.
The
Yeah.
description
If
where
that's public knowledge, yeah.
of the podcast.
Yep. So
Are you
Is
sure?
there
Oh,
is
is
there
that
a
bad?
way to leave it to like put a description?
maybe if they reach out.
Yeah, if they reach out.
If they reach out, we'll send that over to them. That's a good
Okay,
point.
so I'll email you the list, and if they
Yep,
want,
perfect.
or feel free to put it under the, whenever you publish it, feel
Yep.
free to put it in the description and say, because
Oh,
these people all said, yes, please send patients to us that
Okay.
want to
In
want to work
description. In
with us.
description.
In description.
Glad we clarified.
Okay, so there's that, but then Georgie, there's also the fact that you are self-publishing this.
Mhm.
And we've had several people reach out in the past, "Hey, how can I contribute to the funds to support Georgie in this like very transformative work?" So definitely, if you're looking for supplements, support him through IdealabsDC.com.
Yes,
But then, Georgie, is there anywhere that we can send them?
Supplements, because he has pregnenolone, he has progesterone, you have the
yes.
vitamin
I
D.
think that's the best way.
Yeah.
In other words, when it's a fair transaction, there's something exchanged. I'm wary of donations, even though I strongly appreciate them. First of all, it makes me feel like I'm on the hook for something, right?
Yeah.
And I don't want people to lose their money. Most people are not like that. They'll gladly take your money. Maybe not do anything, maybe do something. We'll see. But I don't want. I mean, I feel like I'm on the hook for something, and I should be, right? When
Yeah.
people pay for something. So if that's the case, I would rather be on the hook for giving them product, which is when they buy directly to the company. So currently, the way I'm keeping the cost relatively low by not having a control group, by not having like another standard of care group. Every once in a while, I throw those in whenever the study is very impactful and I want to prove a point. So the cost is still manageable, but I'm starting to get approached by people who want to go and start doing clinical trials, like limited clinical trials. Once it gets to that point, then whoever it has to be a doctor, because they're not going to allow me to do the clinical trials. So it'll be one or more doctors, probably at a university here in the United States. Once it gets to that point, I will just provide the link, the contact information to the doctor or in the institution. I'm pretty sure they take donations, especially if it's going to be like a non-government, non-private industry funded trial. The university is not going to pay for it itself. It's going to say, okay, how do we pay for this? And in which case, I think donations will be very welcome and highly appreciated.
Okay, definitely keep us posted when that happens. But in the meantime, guys, if you're looking for B vitamins, pregnenolone, do you have aspirin?
Not as a separate product. I have it is in the sprayable product called Solbam, which is mostly used for skin protection.
Okay.
But once it gets to, I may have an aspirin like molecule, which is a type of salicylate. So once that happens, you'll be in liquid form. I'll definitely publish on the website and let you know. Currently, not as a separate product that has only aspirin in it.
Okay. So if you're on the market for supplements and you want to support Georgie's cancer studies, check out IdealabsDC.com. He's using that money to self-fund these transformative, incredible cancer studies. But anything else?
IdealabsDC. IdealabsDC.com. IdealabsDC.com is another website.
IdealabsDC.com.
Yeah.
Okay, cool.
Great.
Well, Georgie, we're going to have you come back on in a few weeks because we want to see, there was the tumor size. It went to 0. We'll want to see what has happened afterwards. So hopefully we can email and get that scheduled. But Georgie, I just cannot even begin to thank you for all that you've done. This is just incredible. And I hope that it really starts to help people and wake people up.
It already is. We just need to keep it under wraps until the group is big enough that it cannot be shut down.
Yeah. Okay.
That's all.
All right. Well, thanks so much, Georgie. Until next time.
Good luck with everything in the farm.